So good morning, everyone, and thank you very much for that lovely introduction. I'm very happy to be here to talk to you a bit about some of the research that we've been doing in collaboration with CDPQ, as well as the members of Big Gen Canada, to assess and look for indicators of disease resilience in pigs, ultimately with the goal of being able to select for resilience and producing populations of pigs that are less susceptible to disease. So as Frederick had indicated, one of our initial challenges in this research was to establish a natural disease challenge model. And we're very happy with the work we've been able to do in collaboration with CDPQ in establishing this model. And over the last several years, we've been able to assess the performance and characteristics of about 3,300 pigs that we've taken through this natural disease challenge model from the nursery all the way to grow finish. And you can see from this figure that we assessed a number of parameters in these pigs, in young pigs, all the way through their development until they left the disease challenge model. And as well, we did DNA sampling and did genotyping on all of these pigs. And ultimately, what we want to do is identify phenotypes or indicators of resilience in these animals from these various parameters, but as well look for genetic markers of resilience. And again, as I said, the ultimate goal is to be able to select for more resilient pigs, working with breeding companies so that they can produce better breeding stock that are less susceptible to disease and more productive. So you can see we did a number of assessments, and I don't have time to go through all of them today, but I just wanted to highlight a few indicators that we find promise for and think they'll be useful in selecting for resilience. So one trait that we found quite interesting, and is interesting because it's very easy to measure and it's very commonly measured, is feed intake as well as feeding behavior. So you can see here some data related to feed intake and time spent feeding from a pig that is quite resilient. And what we find is that pigs that are more resilient tend to maintain their productivity. So even though they're being challenged by a disease, they tend to keep eating, okay? So they have good feed intake, and as well, they spend consistent amount of times at the feeder. So this is encouraging for us because it is a heritable trait. It is something that we see consistently in pigs that are more resilient, and it is easy to measure. So we see a lot of promise of using this indicator of resilience because it's easy to measure and measured across a number of phases of production. Another trait that we found quite interesting is as we took the pigs through the disease challenge model, we took blood samples on a regular basis, and one test that we did is complete blood counts, or CBCs. Now, complete blood counts are a very common test done in veterinary clinics, and what we were looking at is immune response. And what we found is that the pigs that are more resilient tend to have a more rapid immune response when challenged by disease. So we see increases in concentration of cells like lymphocytes, as well as an increase in hemoglobin production in the face of the challenge. So I think that CBC is a good indicator of resilience and something easily done, and can be applied quite well in selecting more resilient pigs. The previous two phenotypes are things that we saw in pigs after challenge. We also were looking at tests that could predict resilience or identify pigs that are resilient before they were even challenged. And one test we found quite promising is the high immune response test that was developed at Guelph and is currently being used in the dairy industry for this purpose to detect resilience and predict resilience in dairy cattle. And this high immune response test has two aspects. It has a cell-mediated immune response and a antibody-mediated immune response. So the cell-mediated is a skin test looking for response to antigens, and then the antibody mediated immune response is more of a blood test assessing immune response in the blood. And what we're finding is that this test, particularly the antibody mediated immune response, is a good predictor of resilience. So you can take young pigs, do this test, and identify which pigs are going to be more resilient and possibly have better CBC responses, as well as feed intake behaviors associated with resilience. So this is another heritable trait that we think has a lot of promise in predicting and selecting for pigs that are less susceptible to disease. As I said, we also did DNA sampling and genomic analysis on all the pigs that were run through the disease challenge model, and we identified some very interesting genetic markers associated with specific genes. So the WUR gene, or the GBP5 gene, mutations in that gene have been associated with a reduction in treatments for pigs in the model. So they did not get sick enough in this model to actually require treatment. So that was an indicator of resilience associated with this gene. Similar for the SYNGR2 gene, certain mutations in this gene, pigs would have fewer treatments, but as well they would maintain growth quite well, despite the fact that they were being challenged by disease. So another good indication of resilience. And then additionally, the CD163 is a gene that's been studied before, and we kind of confirmed that it reduces susceptibility to infection by PRRS, okay? And so we have other markers that we are still investigating, but the three here listed here are the ones that show the most promise, but we are continuing to assess other potential genetic markers of resilience. Something else we did for all of the pigs going through the natural disease challenge model was look at their gut microbiome, looking at the bacteria that are growing in their gut. And although we don't think this is an indicator of resilience, it is something that we think we need to keep in mind when assessing resilience, because we've noted that the microbiome and the profile of bacteria in the gut do have an effect on immune response and performance. So the more resilient animals tended to have a certain background or profile of bacteria, a certain microbiome. And as well, we found that particular profiles of microbiome not only affected their resilience, but affected pigs' response to vaccination. So the microbiome in the intestine is something that we need to consider with regards to disease and for optimizing productivity. Some other tests we're still assessing are in vitro tests, tests done in the lab where we can stimulate cells and cell growth as an indicator of resilience from pigs, as well looking at natural antibodies in young pigs to see its relation to health and its use as a prediction of susceptibility to disease and resilience. And as well, we're also looking at gene products in the blood. So looking at if genes are expressed, their transcripts or their transcriptome in the blood, as well as different proteins that could be expressed in young pigs that could predict whether they're going to be resilient to disease or not. And so these are some of the areas we're still looking into and have some promise. But with that said, we're at the stage now where we think we have some strong indicators of resilience, and our next major task is to incorporate these results into breeding programs. So we're now working with members of PigGen Canada to do this. And so working with the breeding companies, we have selection criteria, we have phenotypes that we're interested in, like feed intake, as well as feeding behavior as indicators of resilience, genomic markers that I've discussed, as well as other indicators, so tests that we can help to predict, such as the HIR test. And now we want to start incorporating this into production and working with PigGen members to incorporate these selection strategies into their breeding programs. So we're still, as a part of this, we're still continuing some of the experimental work, continuing the natural disease challenge model to make sure that the markers we have are valid, but as well to help identify other indicators of resilience. We're also starting to look down the production pyramid in the pig industry to look at commercial farms to see if some of these phenotypic phenotypic markers or indicators of resilience still hold true when we look at commercial farms. But as well, we're also starting to look at in depth into the nucleus farms of these various members of PigGen Canada, the various companies, and to make sure that the phenotypes and resilient markers are there and give us the results that we want. So we're now incorporating this and hope to have selection strategies so that the entire pig industry can benefit from this research, and we can have populations of pigs that are less susceptible to disease and therefore more productive. So at this stage, I'd just like to acknowledge contributions of various team members. Dr. Bob Kemp is the chair of PigGen Canada and has done a lot of work in making this research happen. Dr. John Harding from the University of Saskatchewan is a veterinarian who has done a lot of work in making the natural disease challenge model function. As well, Dr. Jack Deckers from Iowa and Graham Plastow from the University of Alberta are geneticists and have done a lot of work in assessing the incorporation of these indicators of resilience into breeding programs. And last but not least, I really appreciate the work that Frédéric Fortin has done to make this research possible. Just wanted to show you this slide to show you the number of partners that we have across Canada and around the world. All of the companies related to PigGen Canada and their support has been very important, as well as support from all of the funding agencies that have made this research possible. So with that, thank you very much for your attention, and merci beaucoup.